The previous lecture left off with this slide talking about codons and reading frames, and the next slide where we saw all of the codons. We started chapter 15 talking about transcribing DNA information to get to the RNA, and now we're going to talk about the details of translating that nucleic acid information that is in RNA in order for the cell to make protein. And that is done by the ribosomes reading the codons that are in the messenger RNA. So that codon is that sequence of 3 RNA nucleotides that specify that one amino acid. The reading frame is how we cluster those 3 in a row together. We talked about how an insertion of 1 or 2 will throw off that reading frame. Later, when we get into genes and gene expression, we will see more about these types of mutations, and how they affect the proteins. For this chapter, we are just going to talk about the proteins. Here's that codon chart, reminding us that there are 64 codons and only 20 amino acids. 3 of the codons are the stop signal that indicate to the cell to end the translation process. And 1 of the codons is the start signal, but that start signal does also specify an amino acid, and it specifies the amino acid methionine. And that reminder that this table will be provided so that if you need to use that on the exam, I certainly don't have all 64 codons memorized, and I wouldn't expect you to have them memorized, but I would expect you to be able to use this table. These are the 20 amino acids, and as indicated on the slide, you do not have to memorize those functional groups, but it is once again reminding us that these amino acids are identified by that specific R group, the functional group that hangs off that central carbon. We saw a larger image of this during the transcription portion of chapter 15, where the RNA polymerase reads the DNA to make the RNA, and they've once again put some T's you think I would have learned in the RNA. These should be, well, those 2 should be U's. Because there are no thymines in RNA. The RNA polymerase has the enzymatic capacity to both open up that DNA molecule, so separate out the strands, and to read the template strand to create the RNA. We also talked about how eukaryotic RNA has to be processed, that the DNA contains both exons and introns. The exons are the regions of the DNA that encode a specific end product, and they exit the nucleus in the form of messenger RNA. The introns are the non-coding regions. They are spliced or cut out of that RNA, and they remain in the nucleus. The exons are spliced together to create the RNA. These have to exit the nucleus, and the translation is going to happen in the cytoplasm where there are ribosomes, and in eukaryotes can also happen on the rough endoplasmic reticulum. The process happens when the ribosome, which has both a small subunit and a large subunit, sandwich over that messenger RNA. And there are 3 pockets in that large subunit called the E, the P, and the A pockets. And they are indentations in that ribosome. Remember that the ribosome is made of ribosomal RNA and protein, so it has a very specific three-dimensional shape. And each of those pockets will fit a tRNA, a transfer RNA, that is the delivery system for those amino acids. This orangey red bar is our messenger RNA. Each of these little bricks is representing a tRNA that is sitting in the E pocket, the P pocket, or the A pocket. Remember that the messenger RNA carries the codons, that triplet ribonucleotide sequence that will tell the cell what amino acid needs to come here. The tRNAs are made of RNA, and they have a sequence that matches, is complementary to the codon. And in the tRNA, that is called the anticodon. So if the messenger RNA has an AUG, there will be a tRNA with the anticodon UAC, because A pairs with U pairs with A, G pairs with C. The second codon on this messenger RNA is UUU, so the tRNA with the anticodon AAA will deliver the amino acid that is specified by the codon UUU. And we see in the P site is where we're building the polypeptide chain, because each time an amino acid joins to another, that is a peptide bond. When we have multiple amino acids joined by peptide bonds, that is a polypeptide. That is our primary sequence. And then it will be released and undergo the rest of its folding through the secondary and tertiary process, and if necessary, the quaternary process. This is in a prokaryote, a bacterium. The green line is the DNA. Remember that prokaryotic DNA does not have exons and introns. If we were to call it one or the other, we would say it's only exons. But there's no nucleus to enter or exit. Just that all of the prokaryotic DNA encodes products. There's no junk or spacer DNA in a prokaryote. So that as soon as RNA polymerase starts to make the RNA, the messenger RNA, ribosomes bind to that messenger RNA and start to translate it. So transcription, as soon as there's enough RNA for the ribosome to bind onto, translation begins in prokaryotes. This cannot happen in eukaryotes, first because the RNA has to be processed so that only the exons are sealed together, and then that processed RNA has to exit the nucleus, and then it can be translated. There is no nucleus with a prokaryote. There's no in and exons. There's only coding DNA. So as soon as we get RNA transcribed, the ribosomes can begin to translate. If you were to build a ball and stick model of a transfer RNA, and it would take a heck of a long time because it's probably a couple thousand nucleotides long, this is what it would look like. It looks more like an R. It's called a tRNA because it transfers amino acids. The anticodon that will hydrogen bond to an mRNA codon is here at the bottom, and at the top we have the amino acid attachment site. This is called a phenylalanine tRNA because this anticodon will hydrogen bond with the codon in the mRNA that specifies the amino acid phenylalanine. Because of this anticodon recognizing the codon for phenylalanine, which must be CUG, it will attach to that amino acid. That means if there are 64 codons, let's see, -3 for the stops, so 61 of which encode an amino acid, then there are 61 different anticodon tRNAs, because each codon has to have an anticodon present on a tRNA, so the tRNA can deliver the correct amino acid. The translation is going to begin not by an entire sandwich and everything coming together at once. Like everything else in biology and chemistry, there's an order and a stepwise fashion that has to be followed. The first thing that happens is the small ribosomal subunit binds so that it covers not only the first codon, but a little bit before it, and it's big enough to cover a little bit after it. Once the small subunit of the ribosome is bound, then the tRNA that has the anticodon for that first AUG start codon hydrogen bonds. And AUG is the codon for methionine. So the methionine is attached to the tRNA. The tRNA is hydrogen bound to its complementary bases from the anticodon on the tRNA to the codon on the mRNA. That is the signal for the large ribosomal subunit to bind. And remember, its shape has those three pockets, the E site, the P site, and the A site. When we bind, this initial tRNA is going to be wedged into the P site. And the P site stands for peptidyl or polypeptide site. There's not a polypeptide yet, but the reason why it sticks in the P site is that the A site, which stands for acceptor, has to be open and available for the next amino acid attached to the tRNA to sit in. So the next codon is UUC. That directs the tRNA with the anticodon AAG to hydrogen bond. Because it's carrying this anticodon, it is carrying the amino acid specified by the codon. Now we've got a tRNA in the P site, we've got a tRNA in the A site. Now we can forge a peptide bond between the methionine and the phenylalanine. This was the first amino acid specified by the first codon. This is the second amino acid, so the peptide bond happens so that methionine is the first amino acid peptide bound to phenylalanine. We see that the peptide bond is happening from here to here because those two amino acids are connected to this AAG tRNA. The ribosome literally ratchets over one codon. That means when we forge this peptide bond between the methionine and the phenylalanine, that left this tRNA empty, and when the ribosome moved over one codon, it pushed this into the E site, which is the exit site. The empty tRNA exits the E site. That movement dragged this one into the P site, meaning the A site is available. We have a CGA codon, so we need a GCU anticodon, and CGA specifies the amino acid arginine. We would forge a peptide bond between the phenylalanine and the arginine. The ribosome would ratchet over one codon, and then this would be the empty tRNA that would exit. This tRNA would have methionine, phenylalanine and arginine attached, it would sit in the P site. There would be an empty A site. This goes through the whole process that we just talked about, where the second tRNA comes in. The ribosomal RNA portion of the large subunit is what has the enzymatic activity to form that peptide bond. When we shift, we pull the tRNA that has the peptide chain into the P site. We have an empty A site for the new tRNA with a single amino acid, and this continues until we reach a stop codon. There are 3 different ones. Any of those 3, if that stop codon is now covered by the A site, we bind it not to a tRNA because it doesn't specify any amino acid. It binds to a release factor that indicates the ribosome should release or come off of the messenger RNA, and the polypeptide chain is complete. We have finished our primary sequence that the cell will then take to complete the secondary and tertiary folding. I have inserted or embedded an animation of this process. As with many textbook-derived animations, the vocalizations, the narration is pretty dry, but it is very accurate. Although those static pictures make it really difficult to see what's truly happening in the translation process, but this does very accurately show that movement, what is happening in the P site and the A site. How does it get from that P site into the exit site? And that means that that tRNA is ready to be used again. So I've embedded that video in this slideshow so that you could watch it, but I have also put the link to this exact video in the module for this week, so you can access it separately from digging through all of these slides. Oops, did not mean to play it. Reminder of what a peptide bond is. These are 2 generic amino acids. The R groups are attached to that central carbon. There's the amino end and the carboxyl or carboxylic acid end. The dehydration synthesis happens between the OH on the first amino acid's carboxyl group and a hydrogen from the second amino acid's amino group that releases water and forges that peptide bond. The reason why we just kept it as R groups is because the R groups do not participate in the formation of that peptide bond. So it doesn't matter which 2 amino acids are coming together. This is always how a peptide bond will be formed. And I end here with the kiwi and a strawberry. Which do you think has more DNA per fruit? Because we're doing that DNA extraction from strawberries or kiwis or whatever other fruit you could find in your kitchen this week. And so you could think about, well, where is the DNA going to be in a fruit? And that would lead you to the correct answer as to which of these has more DNA per each fruit. And that is our very brief wrap up, because as a whole, chapter 15 once again has an awful lot of information. The static images are a bit difficult to interpret, so I do have videos for the replication and for translation so that you can go back and review those as often or as little as you need.